One of the highlights of 8th grade year is participating in a day in the life of Lake Erie and the Niagara River Watershed. This is a project to sample water quality. Join us as we perform these experiments and learn what it means to be a citizen scientist. Today I am joined by Mrs. Feuerman, the 8th grade homeroom and biology teacher. She's going to get us started on our first experiment and explain what this is.
This is a turbidity tube. A turbidity tube measures turbidity. Now you might ask, what is turbidity? It is another word for transparency in some ways. Turbidity measures the amount of sediment that is in water. The clearer the water, usually the better, because that means that the sunlight can filter through and the plants can do their thing, which of course is photosynthesis, providing food and oxygen for all of the organisms in the river. Sometimes clearness is not a good thing because it might mean that the water is too acidic. More on that later. Right, I have filled the turbidity tube up to 60 centimeters. Now what I'm going to do is look down the turbidity tube to see if I can see the black and white Secchi disc that's on the bottom. And I can see it. Ms. Kostin is now going to show us how to measure water temperature using the protocols that we use in Day of the Life. These are globe data protocols. Yes, Ms. Kostin?
Ms. Feuerman, what is globe data?
I will explain that later.
Okay.
What she's going to do is lower the thermometer into the water so that it is halfway down. We will then let the thermometer stay there for about 3 minutes. Ms. Kostin is now going to read the thermometer, keeping the bulb of the thermometer in the water.
We are at 24 degrees Celsius.
24 degrees Celsius. We are now going to wait 1 more minute to see if there is a change in temperature.
Back to sleep.
So 1 minute has passed and...
no change.
Excellent. Now we are going to test the water for nitrates. The presence or the absence of nitrates is very important because nitrates are fertilizers. They cause the plants and particularly algae in the water to start growing and growing and growing. How do nitrates get into the water? Well, runoff from land that is being used for farming that has been fertilized or possibly from factories or something like that. Once the nitrates get into the water, the algae can begin to grow. This can produce something called an algae bloom. Algae blooms cover the surface of the water, preventing sunlight from reaching down to those plants below that perform photosynthesis and provide food and oxygen for all of the critters in the ecosystem. Algae blooms are one of the things that can lead to eutrophication. Eutrophication. What is that? In my words, the death of lakes. I am now going to get some sample water from our site. And this is how we go ahead and test for nitrate concentration. We use this nifty little thing called a nitrate comparator. In order to use it, we have to prepare the water in certain ways. I'm going to go and take the first of the test tubes, take the sample water, and fill it up to the 10 ml line. Of course, as we know from science class, we measure from the bottom of the meniscus. Careful, careful. And we are at the bottom of the meniscus. We will put this test tube into the comparator. We will then fill the second test tube to the 5 ml line with the same sample water. And now we will take the mixed acid reagent, which is one of the reasons why I have to wear all of this protective equipment, and fill it to the 10 ml line. In fact, you can see that there is a color change, and it is clear that they are of different densities as they whirl about. And one drop more. Very good. Now we will cap that, and we mix. And now we wait for 2 minutes for chemistry to happen.
A few minutes later.
Now, I'm going to remove this from the holder, and now we get to add 0.1 grams of the nitrate reducing reagent. So we have this handy dandy little spoon that is exactly 1.1 gram. We have to make sure that it's level. It's very important not to add too much. Very carefully, because this stuff is one of the reasons why we have all the PPE. And now we mix. Okay, I'm going to turn this up and down like this 30 times. And now we wait 10 minutes.
A little longer than a few minutes later.
So now what we do is we're going to slide this bar at the bottom of the comparator. If you look at this, all right, this is our water, this is the comparator. This is a bit pinker than that one. So I'm going to move it one slot over. You will notice that this is pinker than our water. So what we have to do is divide it in the middle. So if we look back at the numbers, we will find that we are between 0 and 0.2. So we are going to record our nitrate concentration as 0.1 parts per million. Now, if you don't understand the phrase parts per million, guess what? We have a lab for that. In order not to contaminate the water at the site, we always make sure to take our waste away with us.
Next, we're going to be measuring pH. This is the measurement of acidity in a body of water. pH is on a scale from 0 to 14, with 7 being neutral. Anything lower than 7 is acidic, anything higher than 7 is basic. The ideal range for lake water is between 6.5 and 8.
So, what does pH have to do with turbidity, Ms. Kostin?
In bodies of water that have higher visibility, they are likely to be more acidic due to pollutants like acid rain that cause organisms to die.
And if pH still confuses you, don't worry, we have a lab for that.
So here is our little canister with the pH strips, and we can see the scale from 1 to this one goes to 12. These are more acidic numbers, the lower pH values. Here's neutral, this kind of yellow, and anything higher than 7 would be basic. We have our pH strip and our sample water, and we're going to dip it in, pull it out, and put it on this white background so we can really see what that value is. So it looks very close to 7. Let's put it next to our 6. I think it's a little more yellow than that. Wonderful. So this would tell us that our pH is very close to the 7 value.
So, now we are going to test for the amount of dissolved oxygen in the water. Dissolved oxygen is important because oxygen is absolutely necessary for life. It is what organisms use during the process of cellular respiration to release energy from the food they eat. And so I have taken a sample of water from our site, making sure that I uncap the sampling bottle underwater and then I recap it underwater so that no oxygen from the air is going to get, or as little oxygen from the air is going to get into my water. So what I'm going to do next is to check my directions and find bottle number 1, which is manganese sulfate. And we are going to put 8 drops of manganese sulfate solution in the sample. 2, 3, 4, 5, 6, 7, 8. Then immediately I'm going to take bottle number 2, which is alkaline potassium iodine azide, and put 8 drops of that into the solution. 1, 2, 3, 4, 5, 6, 7, 8. Then I'm going to immediately cap the bottle. And you will notice a color change, okay, as a precipitate forms.
Mrs. Feuerman, what's a precipitate?
A precipitate, in this case, is going to be solid particles that have formed as a result of a chemical reaction, all right? And I have to wait until the particles settle beneath the shoulder of the sample jar. Now that the precipitate has settled beneath the shoulder of the jar, you're going to uncap it and put in 8 drops of sulfuric acid. 1, 2, 3, 4, 5, 6, 7, 8. Going to immediately cap this. And then I'm going to cap my sulfuric acid to be safe. And now we will mix this until all of the precipitate has completely dissolved, which may take a bit, or even longer than a bit. I have put 20 milliliters of our solution into the titration bottle, and I'm going to take the titration syringe and insert it into sodium thiosulfate. Okay. Then I'm going to pull down the plunger, which is going to allow the sodium thiosulfate to come into the syringe. And we're going to take it all the way down to 0 at the bottom. Right now I have to have it facing towards me so I can actually do this. And we are at 0. Very good. Okay. Now, we are going to add, drop by drop, the sodium thiosulfate. That's the wrong thing. Sodium thiosulfate into the titration bottle until it turns into a pale yellow. This is more of a yellowish brown. So we are looking for this color. So now that we have achieved the desired color, I'm going to put in 8 drops of starch indicator solution. This is going to prompt another color change. That's going to tell us, because guess what? There's starch. 1, 2, 3, 4, 5, 6, 7, 8. Now, we are going to add the sodium thiosulfate until this becomes clear. And then we will know by looking at the plunger on the titration syringe what our dissolved oxygen would be in parts per million. So here we go. Drop and swirl. Drop and swirl. When it happens suddenly.
Mm-hmm.
Okay, is it clear? Or is there color? Looks to me like it's clear. So now we're going to read from the syringe. We are at bottom of the meniscus, 8.0 parts per million, which I believe is a good number.
Now it's time to sample for aquatic macroinvertebrates. I've got my waders on, I've got my life vest, and now I am ready to look for aquatic macroinvertebrates. Let's break down that term though. Aquatic, meaning water. Macro versus micro, where we would need a microscope to look at it. Macro is something we can see with our naked eye. And invertebrates are animals without a backbone. Right now, at my feet, I see an aquatic macroinvertebrate in the form of a crayfish. I'm going to use my net and see if I can catch it. Here we go. Here is our first aquatic macroinvertebrate. A very tiny little crayfish. He's got little pinchers, but they don't hurt because he is so small. And this crayfish is a crustacean, much like a crab or a lobster. And they are an indicator of the biodiversity in this river. So, biodiversity is the variety of life forms, organisms being plants and animals that are in this ecosystem, this environment. And a creature like a crayfish is a very good indicator that this is a healthy environment.
Would you turn it over, please, Miss Kostin?
Yes.
You can see the swimmerettes. Yeah, based on the direction of the swimmerettes, I would guess that it is a male crayfish.
Very cool.
Now don't worry, people. All of our macroinvertebrates will be returned to the environment.
And right now they are just in a temporary home away from home and water taken from the river in this bucket. Some of the other aquatic macroinvertebrates we have in there are these larger dark creatures. These are the nymphs to dragonfly. So dragonflies start out in the water, their eggs are laid in the water, and they are a ferocious predator in any aquatic ecosystem. So soon, this animal is going to crawl out of the water, maybe onto a cattail reed. It's going to shed its skin and molt, much like a cicada, and after it continues to go through metamorphosis, it will have wings and become a full dragonfly. Some of the other creatures in there are aquatic sow bugs, kind of like a pill bug. We have 2 snails on that side of the bucket, some diving beetles, and then a very large beetle larva, which I have not identified without a key or a field guide, but I believe it to be a beetle larva. As far as biodiversity goes for our globe data collection, we have 5 different species. So I haven't spent much time in the river with my waders on, flipping over rocks looking for animals. So I would say the biodiversity at this site is very normal. It's good. As for other creatures, for this environment's biodiversity, we've seen belted kingfishers, American goldfinches, we're hearing catbirds, red-winged blackbirds, so there's a lot of other animals in this habitat using it as its home.
And so, if you look at the data we've acquired today, you will notice that it seems to indicate that the water is doing just fine here at the Stella Niagara Nature Preserve. But
this data is not going to be very conclusive because we've only taken 1 sample. If we were to replicate this sampling process for each experiment that we did today and did it over a course of multiple months, maybe years, Then we would start building a data collection, and we could start making conclusions from that data. But just one data set today is not going to do it.
Doesn't mean a whole heck of a lot. Now, this data, however, is uploaded to a website called the Globe Database. The Globe initiative is sponsored by NASA and NOAA and the NSF and this Department of State. And it allows citizen scientists and regular scientists all over the world to pool their data in order to draw conclusions that really tell us about the world around us.
And citizen science is such an important initiative because it involves the public in scientific experiments. Truly, anyone that asks a question is a scientist.
So, Ms. Kostin, back to data collection?
Back to data collection.